Again, I want to thank Professor Changeux and Professor Corvol for inviting me. My wife and I have been having a wonderful time here in Paris and also in other parts of France, so we're very grateful. And this is the second lecture in this series, and it relies a little bit on what I talked about in the first lecture. But the basic subject is the story of statin drugs, not only how were they discovered, but also how they actually work, which we are beginning to understand at a molecular level. So let's begin with LDL, low-density lipoprotein, which is the major cholesterol transport protein in human plasma. And LDL circulates in blood, and when the level of LDL is high, it attaches to the walls of arteries and enters into the wall, into the tissue of the wall, and it creates atherosclerotic plaques. LDL carries cholesterol, and in the LDL particle, most of the cholesterol is esterified with a long-chain fatty acid and deposited in the core of this LDL particle. And there are about 1,500 molecules of cholesterol ester in every LDL. Back in the early 1970s, 1972, 73, Dr. Joseph Goldstein and I discovered the mechanism by which LDL is removed from the blood. And this slide summarizes what happens. LDL begins life in the liver, and it's made as a precursor lipoprotein called very low density lipoprotein, or VLDL, which contains triglyceride, as well as the cholesterol in the core. And when VLDL circulates through the capillaries, the triglycerides are removed and deposited in adipose tissue for storage or delivered to muscle for combustion to provide energy. And when the triglycerides are removed, the LDL particle shrinks in size, the VLDL shrinks in size and becomes LDL, and then most of the LDL particles are removed very actively by the liver. And the way the liver removes the LDL is by producing a receptor called the LDL receptor, which binds to the LDL and takes it up out of the circulation. So when a person has a large number of LDL receptors, LDL is removed very rapidly and the level of LDL is low. But if, for some reason, the number of receptors is reduced, then LDL builds up in the circulation, it deposits in arteries and causes heart attacks. Now, it turns out that the entire cholesterol homeostasis of the body is under very tight regulation that operates at the level of the cell. And this slide summarizes what we know about cholesterol regulation inside of animal cells. Animal cells, first of all, they need cholesterol. It's a building block for the plasma membrane that surrounds the cell. It is absolutely essential for normal function of proteins on the surface of the cell, and also protects the integrity of the cell by preventing leaking through the plasma membrane. And so the cell has to have the right amount of cholesterol. But if it has too much cholesterol, then the cholesterol will form crystals, and because it's not soluble, it will form crystals and will kill the cell. So cells have devised this regulatory scheme to control the level of cholesterol so that it is not too high and not too low. Cells have the ability to manufacture all of the cholesterol that they need. They can make it from the simple 2-carbon building block acetate through a series of 30 enzymes. But a crucial enzyme in this pathway is called HMG-CoA reductase. And HMG-CoA reductase converts HMG-CoA to mevalonate. This is an early step in the cholesterol biosynthetic pathway. Eventually, the mevalonate becomes polymerized to form the ring structure of cholesterol. But cells have another source of cholesterol. That is, they can produce LDL receptors that bind the LDL in the bloodstream and remove it by endocytosis into the cell. And then within lysosomes, the LDL is degraded by enzymes and the cholesterol is liberated so the cell can use it to make new membranes. And both of these pathways are under feedback regulation. So when cells are deprived of cholesterol, all the messenger RNAs that encode all 30 of these enzymes become elevated. And at the same time, the messenger RNA for the LDL receptor goes way up. And then when cholesterol accumulates within the cell, both of these pathways are repressed. There's a reduction in all of the mRNAs, including the HMG-CoA reductase, and a reduction in the LDL receptor. Now, the knowledge of this pathway that we gained back in 1973 made us think that there might be a way to increase LDL receptors. And that might happen if one had a way of blocking one of the enzymes in this pathway. And since HMG-CoA reductase is considered to be the rate-limiting enzyme in the pathway, the ideal mechanism would be to have an inhibitor of HMG-CoA reductase. And the prediction was that if you could block HMG-CoA reductase and turn off the production of cholesterol by the cell, the level of cholesterol in the cell would fall, and that would cause the cell to make more messenger RNA to produce more LDL receptors, and that would cause the level of LDL in blood to fall. But at that time, in the early 1970s, it was just a theory because there was no inhibitor of HMG-CoA reductase. At this point, a scientist in Japan, very important scientist named Akira Endo, was working at the Sankyo Drug Company. And Endo had read our work and had realized that one might be able to lower LDL levels by blocking cholesterol synthesis. So he set up an assay, a biochemical method, to measure the rate of cholesterol synthesis. And then he screened extracts of fungi. He screened more than 5,000 extracts of 5,000 different fungi to see if any of these fungi made as a natural product made an inhibitor of cholesterol synthesis. And indeed, he found one. And he found several, actually. And he named them in the initial paper ML236A, B, and C. And they were inhibitors of cholesterolgenesis, that is, inhibitors of cholesterol synthesis, produced by a particular penicillin mold called Penicillium citrinum. Now, you notice that this paper was published in 1976, and it was not widely, and it was published in a very peculiar place, the Journal of Antibiotics, which is a Japanese journal. But somehow we found out about this, Joe Goldstein and I, and we realized that Endo had produced just what we needed. Now this is after he found that these drugs block cholesterol synthesis, he showed that the site of action was in HMG-CoA reductase. That is, the drugs were acting to inhibit this crucial enzyme in the pathway. And this shows the reaction catalyzed by the enzyme. It starts with this HMG-CoA, beta-hydroxy, beta-methyl, beta-hydroxy, beta-methyl glutaril coenzyme A. And using two NADPH molecules, it reduces at 2 steps to produce mevalonic acid. And the key point about mevalonic acid is that it can form an internal ester here, which is called a lactone, because of the hydroxyl and the carboxyl group. So it can form an internal lactone. Now, when Endo was able to determine the structure of the molecule that was being produced by this penicillin mold, the shocking thing was that it contained a side chain here that is almost an exact duplicate of mevalonic acid. And in fact, these enzymes are these drugs are competitive inhibitors. They're actually transition state inhibitors of the HMG-CoA reductase reaction. And the first one that he had discovered was called compactin or mevastatin, and these two R groups in this case were hydrogen atoms, and the lactone, although it's shown here as an open lactone, was actually closed. Shortly thereafter came lovastatin, discovered jointly by Endo, but also by scientists at Merck in the United States. And the only difference was that R1 is a methyl group. And then pravastatin, which differs because the ring is open and there's a hydroxyl group. And finally, simvastatin, which became for a while the most successful of these drugs, which has two methyl groups. So the point is, different fungi produce different versions of these inhibitors, and they seem almost designed by nature. It turns out there are 8 asymmetrical carbons here, and they all have to be in the right configuration in order to be an inhibitor of the enzyme. And this is a picture of the different fungi. This is the original one, the penicillium mold that Endo found. You know, the reason he had all those molds was because of the beer industry in Japan. Beer is a major product in Japan, and the beer companies have been collecting all these different strains of yeast to try to make tastier beer. And so the extracts were all there. So we contacted Endo as soon as we read his paper and arranged a collaboration. And this is a collaboration between Joe Goldstein and me and Endo and his associate. And it was the first paper published outside of Japan on the statins. And the title tells the story. It says, Induction of HMG-CoA reductase in human fibroblasts incubated with compactin. And this slide shows the key experiment. When cells were grown in the absence of the inhibitor, and we measured the activity of HMG-CoA reductase, and it was a certain level. But then when we added the inhibitor, now remember what we're doing. We're blocking the production of cholesterol. We're lowering the cholesterol in the cell. And the cell responded by this massive increase in the amount of HMG-CoA reductase enzyme. And the same thing was happening with their LDL receptors. When we were lowering the level of cholesterol in the cell was making much more HMG-CoA reductase and also making more LDL receptors. Now, this enzyme was not active in the cell because the inhibitor was present in the cell. The only way we were able to assay, to measure the enzyme activity, was by diluting, making cell extracts and then dialyzing away the inhibitor. Now, you can also see that when the inhibitor was removed from the cells, in each case, there was a dramatic fall because the cholesterol built up in the cell and the amount of enzyme was reduced. All of this regulation, by the way, is at the level of the amount of enzyme protein. We can't find any evidence for regulation of enzyme activity itself. It's simply that the cell starts making much more protein. Now, the question is, would this lead at a clinical level? would the inhibition of reductase upregulate the LDL receptors and lower LDL. And one of the first clinical studies is shown on this slide. This was done by a clinician named Honeyhockey in Minnesota. And what you can see is, first of all, these are familial hypercholesterolemia. Familial hypercholesterolemia is a disease caused by a mutation in the LDL receptor. And so they have very high levels of LDL because they have a mutation in the gene for the receptor, and they produce fewer receptors than normal. And so FH heterozygotes have one normal gene and one mutant gene for the receptor. And you notice what happens. First of all, they start out with very high levels of LDL. This is, twice normal, three times normal. An LDL level of about 370 milligrams per deciliter. And when they were treated with placebo, nothing happened. But when they were treated with lovastatin, one of the early statins, their LDL levels came down dramatically. This is actually, it shows total cholesterol, but it's all due to a drop in LDL. And then when they were switched to placebo, there was a prompt increase in their cholesterol, and then at a lower dose, they had a lesser effect. So it was very dramatic. This was the first time one could actually titrate the LDL level in blood by adjusting the dose of a drug. Now, the fact that this response requires LDL receptors was shown in this study clinically by our colleagues Scott Grundy and David Bilheimer in Dallas. And they took 3 children who had the homozygous form of familial hypercholesterolemia. That is a much more severe condition. These children have mutations in both copies of the LDL receptor gene, so they can't make any LDL receptors. And when they were treated with a very high dose of lovastatin, notice, first of all, that their cholesterol levels are astronomical, 735. But when they're treated with the statin, in each case, there's no reduction. So in order to respond to a statin, you have to have at least one LDL receptor gene that can be turned on. Now the question is, how does that happen? So about 10 years ago, about 20 years ago, Joe Goldstein and I started to try to understand this control mechanism. And the reason, in other words, how does the cell sense the level of cholesterol, and how is that information transmitted into the nucleus to turn on the genes that make all of these enzymes and the LDL receptor? And it was not a trivial problem, because all of the cholesterol in the cell is contained within membranes. So this is a typical plasma membrane of a cell, and it has phospholipids with saturated and unsaturated chains, and then interspersed among the phospholipids there are cholesterol molecules. And what the cell has to do, what the cell wants to do, is to adjust the amount of cholesterol in this membrane so that it is exactly right. Not too much, not too little, just right. Just right to allow proteins to work. For example, nicotinic acetylcholine receptors, among others. But how does a cell sense this level of cholesterol? How does it measure the level of cholesterol in the membrane? The key breakthrough came in 1993 when a postdoctoral fellow in our laboratory, Xiaodong Wang, purified this protein. We called it SREBP, which stands for Sterol Regulatory Element Binding Protein, SREBP. And this protein turns out to be the protein that controls the messenger RNAs for all of those enzymes and the LDL receptor. And the important part of the SREBP is this part. It's about 400 amino acids, and it's a classic transcription factor. So transcription factors are proteins that are made in the cytoplasm. They go into the nucleus, and they turn on genes to make messenger RNA. And one family of transcription factors has this particular structure: basic helix loop helix leucine zipper, BHLH zip. So in that regard, the SREBP resembles other transcription factors that had been identified earlier, such as MYC and MyoD and others. But when we isolated the SREBP and cloned its gene, we realized that there was something different about the SREBP, because all of the other proteins that are BHLH zip proteins stop right here, right after the transcription factor domain. But the SREBP polypeptide chain continues, and it has these two membrane-spanning helices, and separated by this short loop of 50 amino acids that we call the luminal loop. And then that's followed at the carboxy terminus by another 600 amino acid regulatory domain. And when SREBPs are made, they're not made in the cytoplasm. They're in the cytosol. They're made as membrane-bound proteins, intrinsically anchored within the endoplasmic reticulum membrane. Well, if the SREBP is in the membrane, how does this part get into the nucleus to turn on the genes? That was the next puzzle, and I'll show you, summarize what we learned about how that works. We call it the SREBP pathway. And the key regulator of this pathway is a protein called SCAP, which originally stood for SREBP cleavage activating protein, SCAP. SCAP is an escort protein. Immediately after the SREBP is made, it binds to SCAP. And notice that SCAP has 8 transmembrane helices. So SCAP is an intrinsic protein of the membrane. It has 8 transmembrane helices, and this part of SCAP forms a complex with the regulatory domain of the SREBP. Now, it's crucial for that to happen because SCAP turns out to be an escort protein. And SCAP has a binding site that binds to a complex of proteins called COP2 proteins. And COP2 proteins were identified by others, specifically Randy Sheckman and Bill Balch. And the COP2 proteins are known to be able to bind to proteins in the endoplasmic reticulum membrane and cause them to cluster into vesicles that bud from the membrane as COP2-coated vesicles. So the SREBP binds to SCAP, SCAP binds to these COP proteins, and the COP proteins cause the SREBP to be incorporated into vesicles and carried to the Golgi complex of the cell. And you all know that the Golgi complex is sort of a way station in between the endoplasmic reticulum and the plasma membrane. And when the SREBP reaches the Golgi complex, it is digested by a protease called site 1 protease. We were able to identify and clone this protease, and we found that it is a unique serine protease that is anchored to the membrane of the Golgi by a single transmembrane helix. And this site 1 protease makes a clip in the luminal part of the SREBP. And when it does that, it separates the SREBP into 2 halves. And this half of the SREBP, which contains the basic helix loop helix transcription factor, is still bound to the membrane because it has this single transmembrane helix. At this point, the SREBP is cleaved by a second protease. When we identified this protease, we initially thought it was unique, but it turns out that relatives of this protease go back as far in evolution as archaea. And this is a zinc metalloprotease, and it has the unique ability to cleave proteins within the plane of the plasma membrane, of the membrane. And in this case, it cleaves within the Golgi membrane, and it releases this basic helix loop helix part so that it can travel to the nucleus, and there it binds to sterol regulatory elements in the promoters of all of the genes that are necessary to restore cholesterol to cells. Now the question is, why does the cell go through this tortuous process? I mean, why not just make an SREBP like every other one, a basic helix loop protein, and let it go to the nucleus by itself? Why do you have to make it on a membrane and go through this processing reaction? Well, the reason is because this transport, the requirement for this transport, allows cholesterol to control the whole process. So when cholesterol builds up in the endoplasmic reticulum membrane, when a cell has too much cholesterol or just enough cholesterol, doesn't need any more cholesterol, the cholesterol binds to SCAP, it causes a conformational change in SCAP, and that causes SCAP to associate with another protein in the ER membrane called INSIG. And when SCAP associates with INSIG, the SCAP can no longer bind the COP2 proteins. So SCAP forming a complex with INSIG eliminates the COP proteins, and now this complex cannot move to the Golgi. So there's no processing. And the SREBP that's in the nucleus is rapidly degraded. It only has a half-life of about 20 minutes in the nucleus of cells. So if you don't make any more, the SREBP is in the nucleus disappears, and the transcription of all those genes declines. So this is the molecular mechanism for the feedback regulation of cholesterol synthesis and the production of LDL receptors. Now, I want to spend some time on telling you what we've learned about the molecular mechanism of SCAP, and we've learned quite a bit. First of all, this is SCAP with its 8 transmembrane helices, and it has 3 domains whose functions we know about. And I'll review those for you in the next few slides, but let me summarize. This loop, which is the loop 1 that projects into the lumen of the endoplasmic reticulum, that turns out to be the cholesterol binding site. That's the part that senses the level of cholesterol in the membrane. This sequence of 6 amino acids, methionine, glutamic acid, leucine, alanine, aspartic acid, leucine, we call it malodal. This sequence of 6 amino acids is the recognition site for the COP2 proteins. So this is how the SCAP facilitates movement to the Golgi. And then this domain, which is truncated here, but it's about 600 amino acids, this domain is what allows SCAP to bind to SREBP so that it can transport the SREBP. Now let me show you first a little bit about this malodal sequence. I don't have time to show you all the data. This was published in the PNAS in 2007, but I just want to summarize what we've learned about what happens to this malodal sequence when cholesterol builds up in the membrane. When cholesterol is low in the membrane, this loop 6 that contains the malodal sequence presents the malodal sequence to the these are the COP2 proteins, SEC23 and SEC24. And we can show by in vitro assays that under these conditions, the COP2 proteins bind to this mulatto sequence. When cholesterol builds up in the membrane, there's a conformational change in this loop 6 so that the mulatto is no longer recognized by the COP2 proteins. And we have indirect but pretty convincing evidence that what happens is that in order to be recognized by the mulatto sequence, by the COP proteins, the mulatto has to be at a certain distance from the membrane. And when cholesterol binds, it simply moves the conformational change in loop 6 moves the mulatto sequence so that it can no longer, it's no longer in the right position to bind the COP2 proteins. We know that it's still exposed because we have a monoclonal antibody against this mulatto sequence, and it will still bind to the mulatto sequence even when cholesterol is present in the membrane. So the antibody can find it, but the COP2 proteins can't reach it. And crystal structures of the COP2 proteins show that they are, their binding sites are located at a fixed distance from the membrane, all of which fits with this idea. So, so somehow, when cholesterol builds up in this membrane, the mulatto sequence is moved. Now, what other evidence do we have for a conformational change when cholesterol is added? Well, we've done a lot of experiments with trypsin digestion. And so what we do is we make sealed vesicles, sealed membrane vesicles that are impermeable to trypsin. So this part of the scap is on the outside of the vesicle. This part is shielded from trypsin by being on the inside part. And we have an antibody against an epitope in this region, in loop 7. And then we digest the membranes with trypsin. And we know that trypsin cuts, and then we take the tryptic products, the trypsin-resistant products, and we run them out on STS polyacrylamide gels and measure the size of the loop and blot with this antibody against this loop 7 epitope, so we can see where the cuts have been. And we know that when cells are either in the absence or presence of cholesterol, the trypsin cuts this cluster of basic residues. And in the absence of cholesterol, the trypsin cuts at this position, this amino acid 496, this arginine at 496. But it doesn't cut at 505. And then when cholesterol is added, 505 becomes exposed, and now you get a smaller product because it's been clipped here closer to the membrane. And let me just show you some of the data. So here cells have been incubated in the absence of cholesterol, and then the membranes have been made, and we've digested them with trypsin, and now this is the size of the larger fragment that's produced. But even within 1 minute after we add cholesterol to these membranes in vitro, within 1 minute, you see this arginine 505 exposed. And that's what this lower band represents. So cholesterol, when added to the membranes, causes this conformational change in loop 6, which is the loop that carries the mulatto sequence. Now, what about cholesterol binding? What do we know about that? Well, the first thing we did was, we meaning Arun Radhakrishnan, one of our postdoctoral fellows, was able to solubilize SCAP. First of all, he only used a truncated version of SCAP that stops here at 767. It doesn't have the big part that reacts with it binds to SREBP. So it's just the membrane part of SCAP. And the key happened to be this incredible detergent, phosphocholine 13, which works better than any other detergent in preserving the SREBP and the SCAP structure. And when he solubilized the SCAP and then incubated it with labeled cholesterol, he got saturable binding. But other sterols, also in labeled form, didn't bind. So there's a specific binding site in SCAP for cholesterol. And so where is this binding site? Well, we then started making different fragments of SCAP, and it turned out that the cholesterol binding site was in this particular loop. We thought it would be somewhere in the membrane here, but it's not. It's in this loop that projects into the lumen. And here so we were able to produce this loop as a truncated protein, and we show that it can bind tritiated cholesterol with the same kind of kinetics that the whole membrane domain does, but it doesn't bind a control sterol, 25-hydroxycholesterol. And this is an important experiment because we did competition assays to see what would compete with cholesterol. And we tested all of these 21 sterols. And we found perfect correlation. So all the sterols shown in red were able to compete with cholesterol when assayed in the whole cell. When assayed against the entire SCAP 1 to 8 membrane part. The same sterols also competed when we simply assayed this loop 1 by itself. And then a whole host of other sterols don't compete, and these guys are in the middle in both. So there is, and I should point out that all the ones that compete, all the sterols that compete and bind to SCAP also stop the processing of SREBPs in cells. Again, a perfect correlation. So now what does the membrane domain do? If this is the cholesterol binding part, and this is the part that gets us to the Golgi, what does the membrane part do? Well, it turns out that the membrane part of SCAP is the part that binds to INSIG. And the first hint of that came when we isolated mutant cells. One of the great virtues that we have in our system is that we can use somatic cell genetics. We can take Chinese hamster ovary cells, mammalian cells in tissue culture, mutagenize them, and we can then isolate mutants that have different properties. And one class of mutants cannot turn off SREBP processing. That is, when cholesterol builds up in the membrane, they still process SREBPs. And when we sequenced 3 of those mutants, all 3 of them, 3 independent mutants, all had point mutations, amino acid substitutions in this membrane domain of SCAP. And one of them that I'll talk about is this tyrosine 298 changed to cysteine. And this simply shows the biochemical phenotype. So this is, in this case, we've taken cells, incubated them either in the absence of cholesterol or in the presence of cholesterol. And then we've isolated the nuclei and measured the amount of processed SREBP that's in the nucleus. So when you take cells, wild-type cells, and incubate them in the absence of cholesterol, the SREBP has been processed and it's in the nucleus. When you add even low levels of these sterols to cells, the SREBP disappears from the nucleus because the SCAP can no longer move to the Golgi. But if you have any one of these point mutations and you add even high levels of sterols, you still continue to process the SREBP. So these point mutations make cells resistant to turning off SREBP by sterols. And it turns out that the reason the cells are resistant is because SCAP can no longer bind to INSIG. So this is an experiment in which we used a blue native gel. This is a non-denaturing gel. And we've run out the extract of proteins from membranes. And when wild-type cells are incubated in the absence of cholesterol, and we blot for INSIG, the INSIG runs as free INSIG. It's not complex with SCAP because it's the absence of cholesterol. But when we add cholesterol to wild-type cells, the INSIG binds to SCAP, and therefore the SCAP-INSIG complex moves more slowly on this gel. If you take one of these mutants, the tyrosine 298C, in the absence of sterols, the INSIG is free, but even when we add sterols, there's no SCAP-INSIG complex. And what I'm showing you here for this particular mutant is also true for the other two mutants. So these point mutations in the membrane-spanning part of SCAP prevent the binding to INSIG. So we think then that we understand, at least in broad terms, how the SCAP senses cholesterol. Cholesterol binds to this part of SCAP. It causes a conformational change here, which stops the COP2 proteins from binding. And it does something here to allow this part of the protein to bind to INSIG, which reinforces the trapping in the ER. Now, finally, I just want to tell you how sensitive this system is and why it appears to be working by an allosteric mechanism. So what we've learned to do is to purify endoplasmic reticulum membranes from cells. And we can measure the amount of the nuclear form of, first of all, we can add sterols to cells. Here we're adding cholesterol in a complex with a carrier called cyclodextrin at different amounts. And what you can see here is we're measuring what happens to the nuclear form of SREBP at different times. And when you get to 50 micrograms per mil for 4 hours, the SREBP disappears from the nucleus. And then we isolate the endoplasmic reticulum membranes from the cell and measure the cholesterol content of those membranes. And so what you can see here is this is the cholesterol content. It goes up very rapidly when you use 250 micrograms. It goes up more slowly when you use 50 micrograms. And it doesn't go up at all with 10 micrograms. And this is the scan of these blots to quantitate the amount of nuclear SREBP. And again, nuclear SREBP goes down fastest when you have 250 micrograms of cholesterol. It goes down more slowly when you have 50, and the processing is not inhibited with 10. Now, if we do lots and lots of experiments like that, you can draw a curve. And on the left panel, our experiments were done where we added cholesterol complex with cyclodextrin. And what you can see here is that as the ER cholesterol increases, there's a sharp drop in, this is SREBP processing. So that when ER cholesterol gets to the range of 5 moles per cent, when cholesterol reaches the level of 5% of all the lipids in the ER membrane, there's this dramatic fall in SREBP processing. When the level is lower, the processing continues. When the level is higher, it's inhibited. But the switch operates right around this level of 5%. And that's true whether you add cholesterol in this, carried by this carrier molecule, or even when you add a lipoprotein that delivers cholesterol through LDL receptors, you get the same thing. And you can plot these data in an allosteric sense, and it turns out that the Hill coefficient, the coefficient of cooperativity, is somewhere around 3.5. And you can move this threshold by increasing the amount of INSIG. So this is an experiment which cells had a normal amount of INSIG, and the switch point is about 5%. But when we overexpress INSIG, now the system becomes more sensitive to cholesterol. And we interpret that to mean that there's an allosteric mechanism in place, that that SCAP can exist in two conformations, either the conformation that binds INSIG and has the molodal hidden, or the open conformation where the molodal will recognize the COP2 proteins and INSIG is not bound. And it shuttles between those two. And what cholesterol does is it fixes SCAP in the conformation that doesn't bind, and the conformation that binds INSIG. And the more INSIG you have, the more you push the equilibrium to the left. So what this summary slide says is that there's a delicate balance, that when the ER cholesterol level is less than 5%, the SREBP gets processed. When it's greater than 5%, the SCAP binds to INSIG, and there's no COP protein binding. But all of it works at this very tight threshold of 5 moles per cent. Now I want to finish by going back to endo, because endo made the breakthrough that allowed all of this to happen by discovering the statin drugs. And what I've been showing you now is that the way that the statins work is by lowering the level of cholesterol in the ER membrane and allowing the SCAP SREBP complex to move to the Golgi and the SREBP to go to the nucleus, where it increases LDL receptors. And just to show that this works in living animals, this is hamsters that were either untreated and then blotted with an antibody to one of the SREBP isoforms, SREBP2. And what you see here is that in the untreated hamster, the SREBP is not in the nucleus. It's out here in the endoplasmic reticulum. But when we gave the hamsters one of these statins, the SREBP moved into the nucleus, just as you see here. Now it's all in the nucleus, and that's what's turning on LDL receptors, and that's what's lowering LDL. So finally, I shouldn't have to tell you this, but there have been an enormous number of clinical trials of statin drugs. This meta-analysis formed by Richard Pito and his colleagues at Oxford in England did a meta-analysis of all these studies. And what they concluded, this is of 170,000 participants in 5-year trials. I mean, this is really remarkable. The decrease in heart attacks is directly proportional to the decrease in LDL cholesterol. They found no evidence for a lower threshold for LDL cholesterol. In other words, the lower it goes, the fewer the heart attacks. And in these 170,000 patients, there's been no evidence of significant toxicities or increase in deaths from cancer or other diseases. So, and I want to finish by the way I started. Claude Bernard is the person who enunciated the principle upon which all of this work depends, and statins and endo and everything that we know about depends on the incredible control mechanisms that cells have to maintain the fixité of their milieu interior. That's what the cell is trying to do, and the statins basically exploit this principle for a good medical result. Once again, thank you very much. It's been a great pleasure to be with you.